Зараз всі, у кого будуть запитання, до вас будуть підходити, давати мікрофон. Будь ласка, зараз у нас питання по самій лекції, тобто ті питання, які у вас виникли впродовж цих 45 хвилин, і потім ми перейдемо до більш неформальної частини. Будь ласка, якщо дуже хочете задати питання неформальне вже зараз, то тоді вже так і буде. Але давайте спробуємо спочатку наукові, а потім загальні. Будь ласка.
So this is for questions, yes?
Yeah, it's like...
Now, I'm a bit deaf, so I may ask you to repeat the question after we get it, if that's good. I like questions, so please ask them. And it doesn't matter what the question is.
Будьте сміливіше.
It really does. Good for you.
Yeah, we have a good microphone.
Okay, I have one question. How it's possible to remove some particular genes from the cell?
Кажіть гучніше.
How it's possible to remove some particular genes from the cell?
How it's possible to remove some
Particular genes
genes from
the cell.
When you say equal genes, what do you mean?
particular.
You mean when I think you're referring to the fact that when we sequenced the whole genome, we had 5,000 genes and we eliminated one at a time. Yeah? Okay. So this is, of course, long after the work I described. And what we did is the following experiment. We did it in collaboration with a consortium in South Korea. We arranged it. What you do is you sequence, you have a gene, and then you have nucleotide sequences on the left and the right. Okay? So you can synthesize short pieces of DNA that correspond to the left region and the right region. And we can put in the middle some marker we can select. We then put it into the yeast, and it recombines, these ends find each other, recombine, and replace the gene with the marker that you can select for. So that deletes the entire function. And we did that 5,000 times, and deleted every gene one at a time. Now, many of these genes, or some of these genes, are needed for life. Okay, so if you do that, you kill the cells. So we did it in diploid cells, where you have two copies of the gene, one which remains intact and one which is deleted by this process. Then we sporulate those strains to make two haploids, one of which will be deleted, one of which is not. And then we can study it. So that's the process. We didn't invent that process. It had been done similarly in bacteria and budding yeast before. So we had to develop it to get it to work, but that's the way it does work. We have deleted 99% of the 5,000 genes. 99%. It's the most fully deleted organism in the world at this moment.
І можете піднімати руку раніше, щоб знали, до кого підходите наступного.
Hi.
Hello. First of all, thank you for the fantastic lecture. It was really interesting. My question is, how did you manage to purify the protein? You mentioned CDC2 back to 1970s, 1980s. How did you manage to purify the protein without all these sophisticated
So I
methods?
think the question, if I got it right, was how did we manage to purify the CDC2 protein back in the 1970s and 90s? Actually, I did it in the early 1980s. Okay. I can explain that because I very briefly described it, but I'll do a little more detail, okay? So we had the gene, because we cloned it, right? We put it into a bacterial vector behind a bacterial promoter so that it would be hugely overexpressed in bacteria. We grew up the bacteria, and so you see a new band of protein appearing, okay, in bacteria. We could cut that band out, purify it, because it's a large amount of protein. And then we used that to inoculate rabbits so they'd make antibodies. And we tested to see if the antibodies reacted to that protein. Then we went and made an extract of fission yeast cells, added the antibody, and it found the CDC2 protein in the fission yeast cells, and then we added beads to the antibody, and we could pull them down by centrifugation. So that way we could purify them. Then we had the purified antibody and protein complex, And the question we wanted to ask, if you remember, is did it have protein kinase activity? So we took that immune precipitate, as we would call it. We added radioactive ATP and these range of other proteins that I mentioned to see whether any of them, the protein kinase could transfer radioactive phosphate into any of those proteins. So that's how we did the assay. Was that clear? Good. Once again, we can do it much more easily today. We would just genetically tag it with something else and then bring it down. So we can do this in a week. It took us half a year, of course. Somebody else, I think here? Yeah. Ah, yes. Hello.
So I'm not a biologist, so I don't really understand all the things that you do. And I'm only familiar with the most fashionable ways to alter the genome, like CRISPR and some other stuff. So I really don't imagine how do you change the genome with a mutagen. How do you know that you've changed the right thing? And how do you know that you haven't changed the whole thing?
Well, the first thing I should say is about CRISPR-Cas, which you referred to. CRISPR-Cas allows you to do these sorts of experiments now in all organisms, including humans. The methods I described allowed us to do exactly the same thing in yeast in 1980. So we could gene edit in yeast, which is the reason I worked on it for so long. Only 3 or 4 years ago could you do the same experiments in more complicated cells. So it's what we are doing in all this work is gene editing, but using a different method to CRISPR-Cas. But let me go to your question, which I'm glad you asked it, because it shows the approach you use with genetics. The truth is, we use a mutagen, and it hits everything. There is no selection, unlike gene editing or that we were doing to delete a particular gene when I answered the question over here, which was gene editing. But the way we get around that is we hit everything, but we only select for strains which have a phenotype, a characteristics that we're interested in. So we don't know what gene we're hitting, but we know we've hit a gene that is interesting for the process that we're studying. So it's entirely random, which is the reason I was saying it's a process you can study even though you know nothing about it. Because CRISPR-Cas and gene editing can only work if you know what genes you're interested in. In this case, we didn't know what genes we were interested in. We randomly mutagenize. We screen then for cells that look interesting, and then we define the gene by that process. So in fact, we exploit the randomness to find out how to start. Shall I say that again, because you suddenly looked frightened? Exploit the randomness, I said.
How do you define the gene then? How do you sequence that?
Okay, well, that's the point. We can define it genetically, so we can use genetic crosses to find out where it is on the chromosome. But to find the gene, I had to do the complementation experiment that I described here, taking a gene library and putting it into the yeast. So you have to do all these steps to turn it into something that will work. Today, we do it quite differently. We do it the way I described we've done it. We sequence the genome, we delete every gene by gene editing, which we've done, and then we test each gene one at a time. And that way we can cover everything, and you obviously look much happier doing it that way than the random way.
So thank you. Now I do not understand anything, so...
I'm very sorry. But we can have another go later if you like. Right, don't be inhibited. Yes.
Well, first of all, thanks for an amazing lecture. And, well, you said that you started researching the genes that you accidentally found when looking at those cells with the microscope. You said that those genes, they just differed externally. And after all research you have made, could you explain right now why did those extra genes differ externally from all other ones?
Are you talking about the controlling genes, how they differ?
Yeah.
Well, it's back actually related to the question that we just had. The reason that they were interesting is because what we call the mutant phenotype, what the cells look like, were much more informative. And this is the critical thing with genetics. You have to imagine what cells would look like, or organisms would look like, if they're defective in a gene that you're interested in. Now, you're only imagining, and it may not exist, okay? And we, if you like, could imagine cells dividing faster than they can grow, so they go small. And if they exist, then that would reveal something about the control. But they may not exist. And this is the sort of either the nice thing or the bad thing, depending on your psychology. That you have an imagination, you look for something. If you find it, it confirms your thinking. If you don't find it, then probably your thinking is wrong. So the way you discover the differences that make you interesting is because you've imagined what it would look like and you find it. And this is what makes it exciting. Yeah? Good. And it's related to your question too, action. Anything on this side? Yes?
Thank you for an amazing lecture. I have a question. Like you mentioned that when you try to, that you used 50 proteins to test if CDC2 is a kinase, yeah?
Yep.
And it works on casein, but like casein, I guess it's not a usual substrate for this protein in the cell. Why does it work? Why did it work? Like?
Okay. So you're right. All I did is, if there's any biochemists in the room, all I did is took the Sigma catalogue, which provides biochemicals, and selected 50 proteins. Casein is a milk protein, and it's completely irrelevant, right? It just happened to work at a low level well enough to do the experiment. That's all. So you're asking the question, what substrates are real, I think, yes? You're asking, obviously milk protein is irrelevant, so what are the real substrates for CDC2? What is the CDC2 protein kinase phosphorylating in cells? That's the question, yes?
Actually, no, like,
why did it work? Oh, I'd ask God why it worked. Had no right to work, but it did, and it worked at a low level, and that's fine, okay? You know, so now we have the proper substrate. It works 100 times more effectively. So it was just on the borderline of working, but enough for me to show that it was a protein kinase. But we now have identified hundreds of substrates. I'm now going to ask the question I asked myself. Is that okay? We've done experiments in the recent years which have identified the substrates of CDC2 by what we do is we take a temperature-sensitive CDC2 mutant and we shift to the high temperature. We then make extracts of the proteins before and after the shift, and we analyze them by mass spectrometry using a mass spec, which can measure the molecular weight of the proteins, and we look for those proteins that become dephosphorylated because the protein kinase is defective. And we've used this to identify about 500 phosphosites defining about 200 to 300 proteins. So CDC2 is a massive switch. It activates hundreds of proteins. And we're obviously interested in what some of them do. And incidentally, just to go through it a bit more, we know which ones are phosphorylated with low activity at the beginning of the cycle and which ones are phosphorylated at the end of the cycle too. So we can actually define that developmental sequence. Good.
Hello.
Hello.
You say that sequence process become faster from 1 year to 1 month. Did you invent new ways or technology in sequence or genetic modifier that helps you in your experiments?
My lab did nothing with sequencing. We did a lot with getting techniques to work with fission yeast and modification of genes and did all of those things. Sequencing was invented by Fred Sanger, who was a friend of mine. He got 2 Nobel Prizes, actually, for sequencing proteins and then sequencing DNA. And I did the first gene in collaboration with his lab using his traditional methods. The genes I described sequencing 5 years later were done by improved methods, but still the same method. The methods we use now are a variety of different machines, and we can sequence the entire fission yeast genome overnight, the entire thing, whereas it took us nearly 2 years to do it by traditional methods. We sequenced it in about 1999, 2000, just before the human and fly sequence. But it's got nothing to do with me. I just stole it from other people.
Okay, if I understand correctly, you say that sequence methods are simple, like they start works and they works in that ways now, but better, yeah?
I didn't catch that, actually.
It's в общем, методы секвенирования, они остались такие же самые, только просто улучшились.
He's a lawyer,
Если я
by the
понял
way.
правильно, да?
Sorry? Sequence method is same now, or you have like better version of this methods today?
We have much better methods today,
Okay.
really excellent methods. So a couple of the methods were, the two best methods were invented in England, actually, and now it's just so fast and so cheap. It's just amazing. I told you it took us 2 years to sequence, or maybe 3, the fission yeast genome. We can now do humans for $1,000. It's incredible, absolutely incredible. So I tell you, there's a problem with it. You can sequence so much that people just sequence and don't think about it anymore. They just sequence and sequence and sequence. And there's a sort of, you need sometimes to think a bit. And one of the advantages of experiments going slowly is that you think. When things go quickly, you never have time to think. Or maybe I'm just getting old. That's the other possibility. Now, yes?
Hello, I would like to ask a question about when you used a human kinase library to test whether there is an analog of CDC2 in humans, and after you claim that all eukaryotes have the same mechanisms. Have since there been found any eukaryotes that doesn't use the same mechanism?
Yes, there is. So we started with humans, but we then went and cloned the fly, Drosophila. We cloned from plants. We cloned from budding yeast, of course. I think we cloned from the worms. So we cloned from a range of different organisms and sequenced them and showed they were all the same. Yeah. That's what we did. And now, when you look at the sequence of all organisms, they all have these genes, all of them.
So, like, this mechanism evolved nearly at the same time as eukaryotes appeared, like, type?
Well, I think I understood what you said there. I think what we have to conclude is eukaryotic life. You understand when I say eukaryotic life? Eukaryotic life appeared on the planet probably 1.5 billion years ago. I think that the way in which the eukaryotic cell cycle is controlled was set in place then, and because it's so fundamental to growth and reproduction, it did not change. You couldn't change it. After it had been put in place, you couldn't change it. I already said to somebody that we've identified hundreds of substrates, so if you change this, hundreds of functions would be interrupted. So it's hugely conserved. So it's remained like that for 1.5 billion years. Now, we're less surprised about it now, but I tell you, when we first discovered that, I think people were a bit sort of concerned that we should be controlled just like a yeast, you know? The control of our division and our whole growth and development is controlled in the same way as a yeast. What it also means is that you can investigate how these functions that are important for cancer and other diseases can all be studied to some extent in yeast. It's amazing.
Wow, thank you.
And yeast is cheap, by the way. And fast.
Hello, thank you for your lecture. I really loved it. I have a question. As far as I understand, at the beginning of your experiment, you were inhibiting one gene at the same time, right? But how did you know that it was only one gene, but not several genes responsible for coding same protein? Then you would have to block 2 or more genes at once.
Well, of course, we didn't know there was only one gene. All we knew is that we only discovered one gene, okay? I worried about the same question that you asked, which is why I looked for more and more mutants to see if I could find more and more genes. And I was surprised only to find one, and then the second one, CDC2. In fact, we have now looked for genes which advance cells through the cycle using that genome-wide deletion collection, the 5,000 genes. So we can now look at every one. And the strongest one by far is WEE1. But we do have other genes, maybe about another 10, which have smaller effects. And so it isn't the only gene that behaves that way, but it's by far the most important. We are investigating in the lab today how those other genes work in this system, which is part of the research I'm still doing. Interestingly, can I say something about that? We discovered these sets of genes in the 1980s, and many people have worked on them in many organisms, from yeast to humans. They've all worked on the same genes. When we did this systematic gene deletion collection, the one that we were talking about over here, we discovered another 20 genes or so that we should have been investigating. Nearly all of them nobody has studied. So it shows you that unless you do a systematic study, and this goes back to the point we were going up here, a systematic study looking at all gene deletions, you will only discover part of what is present. And my research for the time being is to try and put all these other ones together, because up until now, they've remained unknown. It's remarkable but true. And this is despite the fact that thousands of people have been working on them. And this is something about science, actually. When people often work on the same things. So if we identified 5 genes that were interesting, everybody worked on them, and then they go and work on something else. But actually, there's many, if you keep going at the system, you'll find that there's many more things to discover, which you need to discover if you're to fully understand it. Is it silent? Good. Hello.
Hello. I have a question. You have already been awarded the Nobel Prize, and what are your next steps in your development as a scientist, if you have a current research?
Well, I don't think I'm going to get another Nobel Prize. If that, getting Nobel Prizes, there's a lot of luck in that. Has to be said. I mean, I got it in 2001, so it's nearly 20 years ago. And since then, I have spent about half my life doing similar things. And I didn't describe many of the things. I just described the first part, not what I've done since then. But I still work on the same problem, really, because it's not yet fully solved. And you need, I think, some people just to keep focused on that, to make, because you get a field, it gets very hot, it's all published in Nature, Science, and so on, and then moves on to something else. And often the field is not yet finished, and you have to re-explore it. And we are continuing to explore it.
And what is your current research now? What do you investigate in detail?
Well, one thing which I didn't explain was that we discovered the first cyclin-dependent kinase. We showed that there were 6 cyclins, so you have 6 different activities. So it was more complicated than I described. Then others working with primarily with mammalian cells and other animal cells described a range of cyclin-dependent kinases, maybe 4 or 5 that are important, and maybe 10 cyclins, and the whole thing got quite complicated. So what we did in several years ago was to see whether we could simplify the system and still get it to work. And it's complicated, and I won't Although I'm trying to simplify it, it's complicated to get there. What we found is that by doing certain tricks, we could re-synthesize an organism which could be driven by only one cyclin-dependent kinase, one protein kinase, one cyclin, and it's perfectly normal. So even though there's 6 in fission yeast, you only need 1. And this simplifies the whole system and makes you focus on what really matters. Because the problem you have with biology is that you can get distracted by detail. And because there's lots of details. And so the thinking here was that if we can simplify the system and yet still maintain the basic process, it will make you focus on what really matters. And what really matters is this very simple model that I finish with the red. That is, that the whole thing is driven by the fact that a single cyclin-dependent kinase increasing in activity brings about the different events of the cell cycle, and you can successfully complete cell reproduction. You can make it work a little bit better by having other CDKs, but they are not crucial. And that is a major step of rewriting how it works. So what do we do now? Now I'm interested in why some proteins become phosphorylated at low activity and some only at high activity. So we're doing a variety of biochemical experiments to look into that. And I'm also looking to see what effect it has where these proteins are located in different parts of the cell, because that's something that nobody's looked at as well. So I'm looking at that as well. I work only with graduate students these days. You know, there's PhD students and postdoctoral workers. I like graduate students, so I can give them slightly crazy projects and they'll do them. You know, postdocs don't. They sort of are rather more conservative. And we have still quite a lot of fun, I have to say. Though it's tough work.
Thank you.
There's another question there, if you're still up for it.
You said that CDC and cyclins are abundant in eukaryotic organisms, but are there any homologs or in prokaryotic organisms like archaeas and eubacterias?
I think you asked what happens in prokaryotes like bacteria?
Yes.
Yeah. Well, it's completely different. Bacteria also have a cell cycle. They don't have chromosomes like eukaryotes. They often have a circular DNA and they only have one origin of replication, if you're a biologist. So they are different. And they have no cyclin-dependent kinases and no control. The control is completely different. So at some stage in evolution, this CDK control network was invented and superseded what happened before, and there's no overlap between bacteria and eukaryotes. There are similar principles in thinking about it, which, in other words, the logic may have some similarities, but the way in which it's executed is completely different. When I mean the logic, I mean the following. If you have a negative feedback control, that's the logic. But you can execute the negative feedback control by a variety of different molecules. So the logic may be the same, the computational logic, but not the way you perform it. Yep? Anybody want to ask the question they were too nervous to ask? Good. Right, in green. Good for you.
Thank you very much, sir. And my question is, what is the practical application of your discoveries?
What's the, sorry?
What is practical application?
What is the practical application?
Medical, perhaps, or biotechnology?
I'm going to answer it with a story, okay? and then I'll answer your question. You are all familiar with CERN, the Big Proton Collider. Before CERN, there was an American one called Fermilab in Chicago, which was also undergoing collisions. And when the physicists were trying to raise money for it, in America, they had to, the director had to be cross-examined by the American Senate. And he was asked a very similar question to the one that you just asked me. He was asked, what good does this project do for defending our country? To which he answered, and it's a beautiful reply, I don't have it exactly, he said, it does nothing to help us defend the country, but it makes the country worth defending. It does nothing to help defend the country, but it makes the country worth defending. In other words, it contributes to the culture, the civilization, to understand these things. The work I describe here does that in a small way, and that's probably the major contribution I've made. However, you cannot understand cancer without understanding this. So what we discovered in itself does not lead to any usefulness in dealing with cancer, but you cannot think about controlling cancer unless you do understand this. So that is the usefulness. Yes.
Hello, I would like to ask, do you think that some kind of blocking CDC2 in cancer cells can stop the cancer, and it can be in some way used to stop cancer?
Well, because CDC2 and other related functions control cell reproduction, many pharmaceutical companies, when we described this, thought this would be a useful target to control cancer. I mean, it was thought to be useful. I discouraged them from doing it because I said this gene controls the cell cycle, cell reproduction in every cell. So not just cancerous cells. So it will kill all the cells that are dividing, not just the cancerous cells. However, and so I never encouraged them to do it. But it has to be said, I really wasn't quite right, because it turns out that although it has nothing to do with what I've done, that certain inhibitors of CDKs and so on have turned out to be useful in cancer, and there's now about 10 clinical trials involving them which are helpful for cancer. But it had nothing to do with the work I did, it has to do with work that was built on it. But you were going to ask something else, yes? No. No. So it does turn out that work built on what we did is probably going to end up to be directly relevant as well to cancer. But the idea that you could use this to block all cell division is never going to work, because you just kill the body if you do that.
But if we try to do it like only in the cancer cells, but not in all the cells.
You could do that, and in a sense that's what you do with radiotherapy, of course. I mean, radiotherapy kills the dividing cells, and by trying to be very focused, and it's more efficient than using a drug. But it just shows you can... I thought about it and thought it wouldn't be useful, but maybe you shouldn't think too much. Sometimes you should just do and see what happens. And I probably stopped it working for 5 or 10 years by being a bit negative.
Okay, thank you.
Можемо переходити вже до питань в цілому мотиваційних, тому тут вже без... I've said that we are ready to go to some motivation questions.
I might sit down for that. For the motivation, I might. Yes, hello.
Hello. When somebody says research, I imagine collaboration between people. And could you point out one or two common features of ideal lab or ideal partner for research?
Yeah, I'll do it after this question. Collaboration is important. It occurs at different levels. I actually have not, I collaborate with people in my lab, obviously, and that's really important. I tend not to collaborate with other groups very much, but I talk a lot to them. I talk a lot. But I work in a slightly unusual way. I'm always working at the edge of something, and I never quite know what we're discovering. And when you collaborate strongly with people, usually you have to be very clear what you're trying to discover and how you're going to do it and how different people contribute, just the psychology of it. Whereas I find that I can't deliver things that are useful to other people because I'm not sure what I'm going to discover, if you understand. But I talk a lot with them, and with my group, I talk to them every day or every other day. So I collaborate very strongly there. So collaboration isn't just, A does this and B does this. It's also to do with just talking and sharing ideas as well. Okay, we're going to go to more informal questions now about anything, and I'm going to sit down for that if you don't mind, because it's less formal. Do you want? Is this working? Yeah. Hello? Can you still?
Hello.
Oh.
Hello.
I'm going to answer another question. Off you go.
So my question is, everyone has bad days, has bad mood, has, I don't know, some broken hearts and so on and so on. But how, in all of these circumstances, in all of these negatives of the mass media, how to stay motivated all the time, how to work effectively all the time as a scientist, and to be a very good person as well?
Well, I think the first thing you have to accept is that the problem you raise is an important one.
Yeah.
I mean, carrying out research is both incredibly exciting, but also quite demanding. I tried to emphasize in that talk the times when it took a very long time of rather laborious work to do things. And the only plus about that is it gives you time to think about what you're doing, because you're doing something and you daydream, and that can be very useful. But the answer to your question, I think, is this. You have to be driven by what you're doing. Now, people get driven by different things. I get driven by a really intense curiosity about the world. My children used to make fun of me because they used to say, "Dad, the trouble with you is you're just interested in everything," And it's sort of true. And I get really interested in just knowing how something works. I get obsessed with it, curious, really curious. And curiosity drives many scientists, but it's not the only thing. Some scientists want to be famous. They want to write papers that everybody refers to. I'm not really like that, but I understand people who are like that, and they get very disappointed if they don't become famous. I frankly don't care if I'm famous or not. I mean, it's a limited interest. Other people do it because they want to do something useful. The question I was asked here. They want to solve cancer and reduce suffering in the world, which is fantastic too. All of these are different motivations, but you have to be strongly motivated. You don't do this career as a career. It's not a job just to bring a salary home or, to keep, because you fail too often. I'll say a bit more. In a university, you often teach and do research. Now, teaching, you should be able to succeed. There's certain ways in which you can do it, you can practice, and you see the satisfaction of people understanding things. Research, you may get no satisfaction for 5 years or 10 years. I mean, this is tough. I mean, my career has been quite successful, to be quite honest, and we've had many advances. Yet even so, there's been years when not much has happened. And something else I'd say, there's not so many researchers who still keep active later in their life. I mean, when you get beyond a certain age, it just becomes too tiring. There's a few people that keep going, but most people don't. So you need to be motivated because you have to be motivated. And most people are motivated by curiosity.
Related question, why cell cycles? Like, there are so many topics in biochemistry, but why?
Thank you for asking that, because I didn't explain that. And I'm going to now. I decided to work on the cell cycle when I was a graduate student. And my project as a graduate student was the most boring one you could begin to imagine. Now, most graduate students think that they are doing boring projects, it has to be said. In my case, it was true. It was really boring. And I thought very clearly, if I'm going to do this for the rest of my life, it better be something interesting, okay? And so then I thought, whilst what I was doing experiments with some machine, a prototype. It's an amino acid analyzer. Like all prototypes, it didn't work properly, okay? So I had to spend my time sitting with this machine to keep it working. The main reason it didn't work is it had too many safety devices. So as it kept going, it would switch itself off because the pressure would build up. And the only way I could get it to work was to inactivate all the safety devices. But then I had to sit with it because if it really did blow up, I had to switch it off, okay? So I was the safety device. And that meant I had a lot of time to think. And I thought about what is an important problem. And I really thought hard about it. And I thought, well, I'm a biologist, right? So what distinguishes living things from dead things? Not dead, but unliving things. And, you know, you learn this at school. You know, living things, you know, they can detect this. They, you know, they're homeostatic. They have a whole load of properties. But one of them is that they can reproduce. Yes? And cells are the simplest example of reproduction. So I thought, this is a fundamental problem, reproduction. Cells is the simplest system. Why don't I invest time into thinking about how cells control their reproduction? And I've worked on, I did that when I was, I don't know, 23, and I've worked on it ever since.
Thank you very much.
Now, if we're moving into a session with more, I wanted to say some more things about what I've done, because it allows you to ask more questions, okay? So I'm a researcher, and that you've heard, okay? And I spend probably half my life, certainly a third of it, doing research still. But I've also done other things, and you can ask me questions about that too. So I have built up research institutions. So I've invented a research council, Cancer Research UK in the United Kingdom. I ran Rockefeller University in New York, and I'm now set up a new institute in London, the Francis Crick Institute. So I can talk about research institutions and how they are run, or rather how they're not run, to be more precise. Okay? So that's one thing we could talk about. I've also done quite a lot of things on the media, making television programs and things. So I can talk a bit about that as well. On science, and I've also done scientific advice for the politicians. So for 15 years I was on the committee advising the Prime Minister in England, in the United Kingdom, and I'm now one of the 7 chief scientific advisors of the European Union, European Commission, and so I was in Brussels last week doing that. So I can talk about politics as well. So a whole range of things you could ask me about, as well as research, as well as motivation, in other words. So over to you on anything.
Hello. Thank you so much for your great job, really. And could I ask you your personal opinion about genetically modified organism? I read that you write a letter, a Greenpeace letter, about genetically modified organism. Can you explain your position?
Yes, I can. So, in actual fact, I'm leading on this for the scientific advisors for the European Commission. I was finishing a report on this morning. and we will publish it in the next couple of weeks. So it's something I think about a lot. I think that the present European Commission's view, which was set up in 2001 on GMOs, is not fit for purpose. It's as simple as that. It was set up for ideological reasons, where the politicians, reflecting concerns of the public, didn't like GMOs for various reasons which were not scientific. They were to do with ideological reasons like playing God, modifying plants, or if you're green, not liking big companies who were using this, like Monsanto, or concerns about taking a gene from one organism and putting it into another, which, as you see, I've been doing since 1985. They didn't really focus on whether these processes were safe or not. They focus entirely on the method being used. Now, if you have a crop that you want to grow in the field, what you have to really ask is that particular product safe for usage? Not how it was made, but what have you made? Now, we now have the completely crazy situation, which I'll describe to you. You can make exactly the same plant by genetic modification methods as you can by random mutagenesis. Exactly the same. In the case of gene editing, you can replace one nucleotide very precisely, but you use GM methods. You can also replace that by random mutagenesis, okay, chemical mutagenesis, which will have many other mutants there. If you do it by random mutagenesis, you don't have to regulate it strongly. If you do it by gene editing, it's regulated much more strongly. The craziness is the actual product is identical. And yet we have different regulations as to how it's safe or not. This is absurd, and we will show it's absurd in our report. The directive that is driving the European Commission's present policies have got to be rewritten, because when you have policy, you have to have good evidence for good policy. Good evidence for good policy. We now have a bad policy because it's not based on understanding and so on. Now, I want to say something else. Why people don't like GM, why the Greens don't like it, is because plants are being made that are resistant to certain herbicides, and high levels of herbicides are being used, okay? But that isn't to do with GM, it's because you can make the same resistant mutants without GM. It's to do with the way agriculture is carried out. So I have a lot of sympathy with the view that you need to think about the ecology and all these things, but you can only do that thinking about the way in which plants are grown and the circumstances in which the agriculture is carried out. You shouldn't focus, and in fact it's wrong to focus on how it's made. So they got the wrong target. So what we will write is saying, yes, you should worry about Roundup and excessive use of herbicides, but you should focus on the product and how it's used and not how it's made. Was that clear? Good.
Well, hello, it's me over there again.
Who's?
It's me.
Ah.
You see? Oh, hello.
I thought we were being communicated to by God again, but no. Okay.
Well, have you ever tried running your own business which has nothing to do with science?
No.
Oh well, then I have a second question. It's going to be rather interesting.
I do get involved in other things. For example, I'm interested in the humanities, and so I'm a trustee of the British Museum. This is a museum in London that you may know of. So I am interested in running other things, but you said a business. I've never run a business.
Okay, well.
Sorry.
And the second question is, do you travel a lot? And
do I travel a lot?
Yeah, and what's the best place you've ever visited, and what do you consider the best place in the entire world to live?
The entire world to live?
Yeah.
Oh dear, I'm not...
If you travel a lot.
Is there a moral philosopher in the room? I think we need a moral philosopher. I do travel quite a lot. but not as much as people think, because I run an institute and I run a lab, and that takes a lot of my time. But I come, you know, I'm here in Kiev. I'm not here very long, just 1.5 days, but I do travel to do that. I don't travel so much beyond Europe as I used to, because it takes so much time. I guess you were asking the question, is it right to travel a lot? And traveling in planes and carbon dioxide, maybe? Was that? No? No, okay, right, not a... Well, in terms of science, let me say something about, there is a movement that we should now just do video conferencing, okay, and not travel. What I think is, I'm not sure video conferencing is a good substitute for real interaction, you know? I think we are having a much more intimate discussion than you can have on a video conferencing. And if you go to a conference, you can talk to people. It's like saying, well, you never have to go to a violin concert or the theatre because you can see it on video. You know? I mean, but it's not the same experience. So my view is this. I think people go to too many conferences. I do think that. Because they just say the same thing. You know, many of my colleagues are going to a conference every week or two and saying exactly the same thing, and they all end up doing the same thing because they're just listening to each other all the time. I think we should go to fewer conferences, but I think we definitely should go to them because it's a different experience. This is a theatre, you know, a piece of theatre in a way, and it's a different experience from watching a screen. So I'm a big supporter of actual human-human interaction and not two-dimensional interaction. Wave, where are you? Hello.
Hello. I have maybe a stupid question, but I think it's really important for young scientists. Have you ever seen that a process which you have been discovering so much time was not a useful thing, not something great, but a usual one?
Didn't quite catch that, did you?
Have you ever seen that the process which you have discovered, was not a great thing, was not useful, but something usual one?
I'll get there in the end. Yes?
You made something, but it wasn't
right.
Great. It was usual thing.
Oh, you mean have we ever made mistakes and so on and made a mess of stuff?
Mistakes and lost your motivation, or sometimes make...
Probably you make some research, and in result it's not great result, but usual result only just information, common result, not something great.
So the answer is yes all the time. Yes all the time, because you know, if you live at the edge of understanding, you will make many attempts to find out things which don't work. And I told you I work with graduate students a lot. I spend most of my time as an amateur psychiatrist because they do things, it doesn't work, they get the wrong results, and they get depressed, okay? And I have to keep them enthusiastic. Then I go home and have gin and tonic myself in the evening to recover. But yes, there is much that doesn't work. Now, you may also have been thinking, well, should that work be published and so that people don't repeat it? My view is yes, but it's difficult to get it published. Also, sometimes it's not quite as simple as that. You know, when you're investigating something, you try different things, you know, like this, and you make a judgment, is this going to actually lead somewhere? And you try something, it doesn't go somewhere, but you're not sure it isn't going to go somewhere, but maybe you think, well, this might work better. So sometimes the preliminary results are not worth publishing because you're not sure if it's good or it's bad. Do you see what I mean? It's less scientific sounding than people think, because you can approach a problem in different ways, and you only really want to follow a way which is working. And if something isn't working, you may not actually show that it's not working completely. So publishing negative results is something which you've got to be sure they are really negative, and not all people are sure of that when they do that. I'm not sure I'm looking at your face that I'm answered your question. Did you want to ask it again, or? Do it in Ukrainian, and then we get a translation here, maybe.
Чи колись ви сумнівались у тому, що ваше дослідження є дійсно корисним, чимось дивовижним, а не звичайною річчю, якимось звичайним дослідженням?
You mean, were there times when I lacked confidence that I was going anywhere? Yeah? Oh yeah. A story. I don't come from an academic background. I come from a working class family. I don't know who my father is, actually. I'm illegitimate. But this meant that I was brought up not in an academic environment. I didn't do well at school. I couldn't get into university. And this meant that traditionally I failed quite a lot early in life. Now, that has a negative, you fail. It has a positive, that if you get through that, you stop caring what other people think of you and that you're failing. And it encourages you to trust yourself if you think you're right. And what I find is, and it is interesting, is now I have students who had never failed anything in their life. Never. They've gone to Oxford, they've gone to Cambridge, they've gone to Harvard or somewhere, and they are all AA double plus, Then they come to my lab and they fail instantly in a month. Instantly. Because now they are in the real world, and it's not just writing good exams. They're struggling with understanding reality. And they find it terribly difficult. Really, really difficult. So you do have to get used to failure. Now, later on in life, we failed often, and I would abandon it if it wasn't working. Sometimes I've made mistakes. A couple of times I've never published anything that was completely wrong, but twice I submitted something that turned out to be wrong, and then I discovered that it was wrong before it was published, so I could bring it back again. So I have made mistakes, and you will make mistakes, and it's not evil to make mistakes, but you have to be honest when you've made a mistake, and that's fine. And I think we shouldn't get hard on people who make honest mistakes. But the answer is yes, I make mistakes. Yes, I sometimes lacked confidence. I couldn't get a job either. I mean, not only couldn't get into university, but for ages I couldn't get a job as a lecturer or professor and so on. I mean, it all worked in the end. I mean, so it was fine. Not as straightforwardly as you might think. Good.
Okay, hello. Thanks for your lecture and for all your answers. And I would like to ask you two questions.
Yes.
Firstly, about thinking through your results and thinking through your work. You said that in the past there was like 1 year in order to, so you can just think through the work you do, and now there is new technologies that gives us extremely quick results, and we can't do this thinking as we did. And the second one is...
Yes, stop there. Let's just stop there, and then you can ask the third one. So I think you're asking... Oh, well, no, you have just about this, when you are getting very rapid results.
Yes, and you're like, you have these results and you don't know what to do with them.
Yeah. The work I've done never seemed to be rapid results, actually. It was always rather slow. That isn't the case in all research that people do, but in our case, it was rather slow. And I think it has something to do with the fact that we're very careful. sometimes, this is an interesting experiment, interesting question you're having. In biology particularly, you can be working towards an objective, this objective here, okay? And you're doing experiments. And you have an idea that may be sensible. And I have always had the view is that you should hate your own ideas. You should try and destroy your own ideas, okay? And so if I'm working with somebody, a student in my lab, we constantly are trying to destroy the idea. Now, sometimes if you stop earlier, you can write quite a good paper. But I keep going, and sometimes this has been destroyed, and we can't publish anything at all. Now, this isn't good in a sense for a career. It's good for doing good science, but it's not good for a career. And so you've got to be very careful about that. But it does mean, science is the pursuit of truth. You're not always going to get it right, but it's the pursuit of truth. So you should test your idea to destruction. And some of the problems that we have with the literature isn't, particularly in the biological, biomedical field, is because people are publishing too quickly before they've tried to test it to destruction. Physicists are better at this, I think. I mean, they work in a clearer framework of understanding, and I think it doesn't happen so much. But in biology, it does. Does that answer the question?
Yes.
Good. Second question.
And the second question is about the qualities that young researcher, young students have to develop in themselves in order to be a great researcher and great scientist.
Yeah, the qualities to become a great researcher. Well, first thing, there are many different ways of working, and there is no single way of working. And that's important to realize, because some people want to do very adventurous things. Some people want to do things that are very thorough but more conventional. So, for example, sequencing fission yeast and then the human genome was really boring, but very important. And I don't normally do that sort of thing. I did it because we had to do it. But if you are working in something that is very uncertain, then that's more exciting, but you're going to fail a lot. I sometimes use a military metaphor. Imagine war, and you have trenches, and you're advancing just meters at a time, but they are important meters. Or imagine somebody's parachuted in 10 kilometers on the other line. You have a different way of working, and that's important. Okay, so the way in which you do science varies from one to another. The qualities which I think are common are you have to be extremely motivated. That goes back to the first question. There's no point in doing it. Motivations can be different, but you have to be motivated. You have to recognize science is a pursuit of truth. I mean, it's a high calling. It isn't just producing publications. That is, of course, a measure of career, but the real objective is to try and understand something. And you have to always keep your eye on that. And if that means you can't publish something because it's not done, didn't come out the way you have to live with that. Unfortunately, the way decisions are made about careers, they don't always recognize that, but they should. The leadership should. So you have to be aware of that. Third characteristic is you need to be self-critical. People like their ideas too much. I've already said that. You have to be self-critical, because that is a correction for mistakes if you're self-critical. Notice I haven't said you've got to be bright, but it's helpful if you're intelligent, okay? So good if you're intelligent, but it's not enough to be intelligent. You have to be intelligent with a work ethic and always recognize that everything you do is dependent upon good data. I get very smart students coming through. If they produce rubbish data, they can't do anything. I always remember one of my graduate students who's now a full professor in University College. His first experiment did this, okay? I forget what it was, but it did this, okay? He was very smart. So he had an explanation for everything, right? So he spent an hour describing why it went up, then it went down, then it went up, then it went down. And at the end of it, I said, why don't you do the experiment again and see if it's the same? And of course, it wasn't, okay? It was just random. So you have to have good data and rely on good data. What have I left out? Is there something you think that I've left out? Microphone.
About self-criticism.
Self-criticism, yeah? Self-criticism is really important, but you can't. But you've got to be subtle about it. If you're so self-critical you don't do anything, that's a mistake too. So you have to have an energy to do things, but to be self-critical about what you're doing. Yeah, so that's a good point. I sometimes use the following language. You have to have an in-depth understanding and knowledge, but have a superficial vision of what else is going around you. And a danger for researchers is just to be focused on their one little thing. And there's a very important lesson there. I call it running on railway tracks. When you think about a problem and you have an idea how it's working, you start running on a railway track. You can only think about the idea in one way. And there's something about that which inhibits other explanations. I don't know what it is, but it does. And one of the most difficult things in doing research is to jump the tracks into other ways of thinking, because many times in my life I've been doing this and thinking there's no other explanation. And then suddenly you realize there is. So you have to play tricks on yourself to jump the tracks. What tricks can you play? One thing when I think that's happening in the lab, I tell people to go on holiday. Just stop thinking about it, go away. Or go and walk up a mountain or something, you know? And just forget what you're doing. Just switch it off. So when you switch it on again, maybe you'll come onto another railway track. And this is all to do with being self-skeptical, because if you're so sure your idea is right, you just run along that railway track, and you have to be able to jump railway tracks. Does that? Yeah.
Hello, thank you very much. And I want to ask you, when you start your research, what goals did you have, and what goals should have young scientists? Maybe you want to do some great inventions by yourself or something like this, or create some new treatment of cancer.
I think I got it, but you...
The question is about what goals should have young scientists, and what goals did have you?
Goals in the beginning of science.
For me personally, yeah.
Yes, for you.
I tell you what I was thinking about. You know, when I was told you the story about why I started working on cell reproduction and, you know, the properties of life. What I felt then was that I wanted to do research. I did want to do that. But I felt strongly this, that a couple of you have asked what was useful about what I did. And what I thought at that time in my 20s was, if I'm really very good, then I can justify working on whatever I think is interesting. But if I'm only good, then I must have an objective of being useful. But if I'm very good, then perhaps I will find something that will be even more important. Otherwise, maybe I should just work on an infectious agent, malaria or something, and do it in a good workmanlike way, but not be... And so my goals early on in life was to test whether I was really good or not. And if I wasn't, I would have gone into a more conventional way of working. I never thought that I wasn't thinking I'd be very hugely successful. I used to think if I was really, really good, I'd become a member of the Academy, the Fellow of the Royal Society. That was my peak. And I did get that, but I did beyond it. I became president of the Royal Society eventually, for example. But I didn't have a goal, a sort of goal beyond that. The goal was to find things out. But mainly, if I was very good at it, I would trust my judgment. If I was less good, I'd do something useful. There are several questions arising here.
Hello.
Wave, where are you? Ah, hello.
You said that you make science consulting for politicians.
Yes.
What is the most ridiculous case of a misunderstanding with politicians in some science questions?
What's been the most... Well, you know, my experience... I didn't work with politicians in the United States. When I worked for 7 years, 8 years in the United States, and I didn't do that there because I didn't understand American politicians. And I understand them even less now, actually, of course. I do understand European ones and British ones better. British politicians I worked a lot with, and although I might politically disagree with some parties, which I do, on the whole, I found that they were pretty sensible. I'm not that critical. What I find in working with politicians is something which is obvious. What happens to many scientists? They only go and talk to politicians when they want to complain about something. You know? And they're usually complaining that they don't have enough money. That's the usual sort of thing. Now, if you think about relationships, if you want to be friends with somebody and you only talk to them when you want money out of them, you don't get very far, you know? So my approach there has always been to make friends with them and talk to them as if they're human beings and explain things to them and don't demand things of them. And then when you do want something, they're more likely to listen to you. And if they trust you, then most politicians, certainly in the United Kingdom, were okay. In Europe, I found that isn't quite so the case. I think that we talked about GMOs, for example, and the GMO thing is very disappointing to me because it isn't rational what has happened there. What's good in Europe is that they will say they take science seriously, but they haven't quite got it in their DNA yet. So, and that is proven more of a struggle. I was in Brussels last week, for example, to talk about GMOs and the report we're writing. And the civil servants just didn't want to talk about it because they said it will all be too difficult. We know it's crazy, but it's all too difficult. And that is just cowardly, you know, that's just cowardly. But the politicians, so, for example, the Commissioner for Science and Research is absolutely in Europe, who's Portuguese, Carlos Moedas, is absolutely on board. But if you go to more senior politicians, they don't want to be disturbed. And so I haven't answered your question about what was the worst thing, because actually I've not had too bad an experience, to be quite honest. I mean, I couldn't cope with Trump. I mean, the guy is crazy and takes no notice of evidence and science. I mean, that would be impossible to deal with, you know, utterly impossible. Okay?
Thank you very much for your election. I have a question about your working as a director of research institutes. And can you say what was the most difficult challenge for you, where you was like a boss in there?
Well,
yeah, thank you.
I think I have very strong views about how you carry out good science, and good science is carried out by good people. So the key is to identify accomplished people who can do things. The key is not to direct them and say you must do this or this or this, but actually identify good people, often young, who will create the new discoveries of the future. And the problem I've had is that the administrators who control research funding just don't always understand that. They talk a lot about translational objectives, that we have to do this, this, and this. And they don't understand that, yes, you do that when it's close to applications, but if you're trying to discover things, it's completely counterproductive. So I have constant disagreements with the administrators because they always want to control what you do. They want to use ways of measuring what you do which are not very effective. And I'm okay with the leadership, but it's beneath that. Those that sort of run the grant committees and things like that, they hate me, actually. They hate me, I can tell you.
Okay, and a second question is, do you see any global problems in world science? Maybe there are, and like...
Do I think global problems can be tackled by science? Is that your...
No, like, not tackled by science, but also in, in science world, or there is no...
Oh, the global problems in the science world.
Yeah.
There's some great and interesting problems, I mean, like in physics particularly, I mean, such as dark matter and dark energy, which are just extraordinary to me, that we've had 400 years of high-quality physics, and 85% of the world, we don't understand what it is. I mean, is that amazing? I mean, almost spooky, So that's clearly a global problem, I would say. There's 85% or whatever it is out there that we don't know. In biology, we tend to deal with smaller things. I mean, we have one or two great ideas like Darwin and evolution and natural selection, but we deal with more details. I actually carry out research in biology that is a bit more, less detailed and more trying to understand some bigger picture. But my view about identifying big problems in biology is the same answer to your first question. Identify somebody young who's really good and let them find the problems for you. And that's how I run the institutes, by the way. I simply hire younger researchers. I don't care what they work on, if it's interesting, and let them loose.
Thank you. And like a summary, do you have some special advice for your Ukrainian colleagues, your Ukrainian science?
Ukraine, from my understanding, has a strong tradition in science. And I think it should build on that, because the future is dependent on science and science discovery. And Ukraine has been very strong in the physical sciences and mathematical sciences, and also engineering, from my understanding, too. And it now has an opportunity perhaps to be freer, and it should invest in that area and invest in people, young people. And I think it probably recognizes that, but it does require resources. It does require the will to do it. But I think Ukraine can play a big role in Europe. It's a big country. It's got powerful traditions. And really we need to embrace countries like the Ukraine more closely into Central Europe to make this bigger mass of science. Because if you think about the big critical masses of science, you have the United States, which is possibly on the decline. You have Europe, which is still trying to invent itself, really, but which is overtaking the US, in my view, and can get bigger. And then you, of course, have China and the Far East. And these are the three great blocks. And my view is that European scientists should work closer together, collaborate well, talk to each other, and they will become the most powerful bloc in the world. And I think Ukraine should contribute significantly to that.
Hello. Thank you a lot for coming and for spending your time on us. Actually, my question is connected to a previous answer. How, of course, in your opinion, how is it possible to develop science in a country, for example, Ukraine, when there is almost no support from the government or any kind of involvement of it?
Well, I think you need to have recognition in the country that science is important, and that's a political question. Once that's happened, then there's several obvious steps. One is to have an educational system that promotes that. I actually think, from my understanding, the Ukrainian education system is strong. Traditionally, I think more in the physical sciences, as I've said, but it's strong. And I think that's critical. And then you have to build a science infrastructure. I mean, it has to be seen as an important endeavor for the country. And that does require political leadership, because if you're in a country and you're worried about the economy and worried about public transport and housing and all these things, science can look a little bit into the future too much. And so you have to have leadership that recognizes that the future is science and it needs supported. It's not that expensive. You know, we're talking about spending in government spending of 1 or 2% a year, so it's not hugely expensive, and it makes an immense difference for the future. It's political will, in other words, political will. And scientists have a role to play that. Our scientific leaders have to project that, and they have to have a big vision for it, and they have to have a responsibility to drive it. I think the mistake is when scientists spend their whole time just in their academic endeavor and do not engage with the public and politicians. We have to do that. Sometimes I use a metaphor that you have to have a license to operate scientists, but you have to earn your license. You have to earn your license all the time by engaging with the public and engaging with our political leadership.
Hi, sir, I would like to ask...
Hi.
I would like to ask you if, in your opinion, Nobel Prize is always fair. Because, for example, DNA-related researchers, people get their Nobel Prizes like in 4 years. So, for example, Watson and Crick and Kornberg with the DNA polymerase. And, for example, Elizabeth Blackburn, she had been waiting for 20 years. And the most known example, Einstein, because he got a Nobel Prize for the photo effect and no Nobel Prize for the theory. So, what's your opinion? Thank you.
Well, I sort of think, you know, no prizes are completely fair, to be honest. I mean, by the very nature. I always say that for every person who wins a Nobel Prize, there's probably 50 others who could. I mean, so it's not a question of I don't think the Nobel Committee makes too big a mistake. I do think that. But I do think that, or not in recent, not the last 50 years or so, but I think that there are many more who are of the equal standing to a Nobel laureate. So I think that it's a mistake to think that the Nobel laureate is above everybody else. There's many others who are similar to them. But to win a Nobel Prize requires certain characteristics which you need to be aware of. The discovery, and it's given for a discovery, not for a person, a lifetime commitment. It's given for a discovery. It can't involve more than 3 people. If it involves more than 3 people, they can't sort it out. And it has to be clearly supported by experimental evidence. So it's no good if it's just a theoretical, for example. And you have to be certain that you've got the right 3 people. Now, putting all of that together means that there's only certain areas that will get rewarded. Now, there are certain areas that can never get rewarded because they don't fit something, even though they're major achievements. Now, people criticize the Nobel Prize for not doing that, but I don't think a single prize can actually cover all of that. And rather than complain about the Nobel Committee, which I think does a pretty good job, there's these other prizes that get invented. You know, the Breakthrough Prize is the latest one that you may have come back, which is worth a lot more than a Nobel Prize. And they could invent a way of working that could cover some of the areas that Nobel doesn't cover. So, for example, they don't have to be limited to 3 people. They could have teams and so on. So I wouldn't criticize the Nobel so much as criticize some of the other prizes for not, instead of just copying the Nobel, which is what many of them do, of not inventing different ways of dealing with it. Your general question is it unfair? Some people wait a long time. Yes, it is unfair, but I mean, it's every prize is unfair. You know, it's like a book prize or something. There's always unfairness to it. The mistake is to think that this is the only thing that is worth something. It isn't. There's other people who are doing just as well. So the mistake really is that Nobel has got this aura of being the great thing. And that's the mistake, really. Because if it didn't have that aura, then you'd get different prizes given to different people.
Ми маємо 2 ще запитання. Це от 3 запитання. Останні 3 запитання. 1, 2, 3. Дуже коротко. Добре, нам треба завершувати. Будь ласка.
So, for example, I'm here. Hello? Hello? Hello?
Ah! Maybe I can make more sense this time.
Sorry?
I'm hoping I can make more sense this time. You remember, I didn't convince you on your last question.
So, for example, I'm doing some scientific research, and I'm getting...
What sort of research?
Scientific research,
yep.
And I'm getting negative results.
Yes.
What should I do if nobody wants to publish it?
Yeah.
Where do I go? And what do I do?
Yeah. Well, first of all, I have sympathy. I know that isn't much use to you, but I have sympathy. Negative results are positive. I'm trying to reassure you. If you've done the work well and it's negative results, you've contributed something. So for your own confidence, if it's done well, it's important what you've done, okay? It doesn't help you with your career, me saying that, but it might help your brain, mind, you know? Because if you, and I tell this to my students who get negative results a lot, if a negative result stops you having to think about certain things in a certain way, you've made a contribution. You've cleared away some of the stuff you shouldn't be thinking about. So don't lose confidence. Don't lose confidence. Now, what do you do about your career? That's what you're asking, yeah? Well, if you apply to people running an institution who are serious people, they will evaluate what you've done. But I have to admit to you that it's not always the case that they're serious people. So if you are exposed to an interviewing panel and they get inside your brain and your brain is a good one and you've done good work, they will see that. And then the negative result may not matter so much. However, the real problem is getting in front of the interview panel. Because if it's negative results, and then they may just look at the paper or look at, or there's no papers, it's more difficult. And that is difficult. You have a supervisor or supervisors who advise you, who can write reference letters for you. You. Yeah. That is really your way forward. If you have a supervisor who can explain what you've done and why you've got negative results, that's the best way to go. Because they can say, this guy, I don't know if it's true, by the way, but I'm saying, this guy is really good, has done A, B, C, and D. It didn't work, but he did it well. It worked, but it didn't give the result we expected, but he did it well. This guy is worth investing in. And that may work. So you should take your supervisor out to dinner, maybe.
Hello, can I ask one question? Are there some actual researches in biology where physics is needed, physics science, that can collaborate to sciences on the edge?
Right, go on.
Ви говорите про колаборацію між біологами і фізиками?
Are there some actual researches in biology in which physics, science, and achievements of physics is really needed? Can you talk about some examples of this nowadays?
So often advances occur in between subjects, which is the point you're making. So in my institute, for example, we've taken a special interest in engaging physicists, chemists, mathematicians, computational biologists, engineers, and that is aimed at getting that level of expertise and that level of thinking into studying biological and biomedical problems. However, it isn't quite as straightforward as that, and I want to explain why. Let's take medically trained people, biologically trained people, and physics trained people. They are all trained in different sorts of ways. And getting them to work together is actually not so easy. And I just think about it. A medical doctor is trained to make decisions on limited information and works in a way which is a strict hierarchy. And if you're a patient, that's exactly what you want. You don't want to be treated by somebody who's age 30 who's had no experience. You want to make sure that they are fitted into a hierarchical system. You agree. And you want them to make a decision about what you should do and how you can be helped. Now you take a scientist like me, a biological scientist. If I'm faced with data which I don't understand or which is not clear, I will always say, well, I'll do more experiments. You know, that's what happens all the time when we're discussing things in how to run the institute. They always say, well, we want more data. You know, it's a typical way. And also, they don't respect authority, scientists. They don't like hierarchies. And so you have a completely different culture of medics trained in one way, scientists in another. Then you have physicists, of course, who are much more respectful of theory and great thinking, and biologists who are much more empirical, and we expect them to work together as well. I always remember when I talked to a physicist about the first results that we had in a genetic network, and I remember him saying, well, you could get it to work with 2 genes. Why have you got 5? And I said, well, actually, because there are 5, you know. But his thinking was such that you should reduce it to the simplest possible system, because that was more elegant, okay? Now, I've given you examples of 3 different cultures that you have to get to work together. So that's more difficult in getting collaborations and across than just simply asking a physicist to help. We need sociologists, probably, to tell us how to interact with different people. And I found that a bit of a problem in trying to integrate the physical sciences and the medical sciences into an institute. For that reason.
So, hi. Sorry for my English. Do you think about in the world there are other people who can use CRISPR-Cas technology like a biological weapon? Maybe today exist plasmid types who are very stable in the environment and big pharmaceutical corporation can use this method to increase number of illness cases for make more money. It's a very common situation.
Well, I think you're asking a question about the use of scientific advances in things that are not good for humankind. And you mentioned CRISPR-Cas. I sort of think we have probably enough biological ways to kill people already, you know, with just natural pathogens and so on. And so if somebody wanted to use biological warfare, it's already pretty dangerous, if you see what I mean. But I think you raise an interesting point about which I'd like to comment on, which is this question of discoveries which can be used for the public good and for the public detriment. And I do have views about that, in that I think we should not inhibit discovery of any sort. When you come to applying a discovery for a particular purpose, so it's more at the applied end, then I think that if there needs to be in a proper democracy discussion about how it's used, and that could be for biological or other chemical or nuclear weapons, it could be for medical interventions like CRISPR-Cas in medical interventions or stem cells and so on. And the best way to deal with that is to have a healthy democracy that allows discussion of these issues, of which the scientific community have a special responsibility to inform the public and society what the issues are, but don't have a special responsibility for deciding it. They have a special responsibility to inform, but the decisions should be democratic and wider ranging. So my view of it is this, that you need a healthy science discovery, but the applications of them are best considered in a healthy democracy. Even in a healthy democracy, of course, you will have people who will have different opinions. I mean, and if you have a strong feeling against nuclear weapons or nuclear power, then you have to go with your conscience, of course. But basically, I think it's a healthy democracy to discuss these issues that is critical.
Thank you.
So another question, I would like to ask you about your position on GMO copyright, because when copyright in GMO, so like patents.
Oh, gene modification in general, yeah.
Yeah. So I mean, like, you almost never design a new protein from scratch and then translate it to gene and insert a gene. So you take existing proteins, existing genes for them, and move them to another crops. So like, but. Also, it takes a massive effort to do this and to do it correctly, to pass all the trials. So, like, it should exist, but if it's too strict, it also could lead us to some kind of dystopian society where you couldn't grow anything without permission of some big corporation. So, what are your thoughts on, like, exact degree of strictness of copyright needed in this area?
I think the critical thing that's needed is a focus on the product that's being made. And the reason I emphasize that is the same answer I gave over there, that if you focus on the way something is made, you're asking the wrong question. You have to ask, is the product safe that's being made in every sense? Is it safe for human health? Is it safe for the environment? Is it safe for other animals and so on? And that isn't the question that's being asked by those that oppose GMOs. They are simply opposing the way in which it's made. And that is a fundamental flaw for good policy, in my view. Because you want to be, if you have something a product that you're using, you want to know if the product is safe. And that's the critical issue. And to focus all the time on how it's made is just a distraction from it. Now, the European Court of Justice ruling, I don't know if you know what I'm referring to, they've just made a ruling, which we're going to oppose, in this ruling, in our analysis. They talked about the fact that GM methods were too fast to be safe. That's what they said. Too fast to be safe. Now, what's going on in their heads to say it's too fast to be safe? That is a way of saying that any new discovery that can help health or something, if it's fast, it's bad. They have locked themselves into a sort of, done in a fast way. They're seeing that as a bad thing. They've just got it wrong. Seriously wrong. And we shall show it seriously wrong, I hope.
And finally, like summary, can you give some 3 advices?
3 advices?
Yes, to this.
And
guys.
Advices about what?
About life, about science, about everything what you want.
Okay. Well, look, the first thing is you do have to be happy. And if you're going to be a scientist and it makes you unhappy, that's a bad thing. So only do it if it keeps you happy. So that's the first thing. The second advice is, if you do science, you're unlikely either to get rich, okay, or famous. So if you want to be rich and famous, I wouldn't do science, okay? And thirdly, if you like it, if you're curious, if you've got patience, it's the most fantastic thing to do. That's my 3 pieces of advice.
Dear friends, on behalf of the name of our rector, academic Leonid Gubarski, I am honored to present this medal award of our university to Professor Sir Polom Ners. Of course, this is not a Nobel Prize winner medal, but I believe that in future it will be equal.
І для фінального слова хотіли б запросити нашого партнера, друга президента Європейської молодіжної академії наук, який підтримує серію лекцій майбутнього, Олесь Довгий. Будь ласка.
I would rather stay in Ukrainian. Друзі, тому що це більше до вас, ніж до нашого поважного лектора. Коротко хочу сказати. Одна відповідь на запитання про те, що треба зробити для того, щоб стати чи Нобелівським лауреатом, чи видатним бізнесменом, чи просто успішною людиною щасливою. Щастя в будь-якому випадку всередині вас, і воно, мабуть, не стосується таких викликів, як Нобелівський лауреат, не від цього залежить. А те, що стосується чогось надзвичайного, то я переконаний, що це лежить не в ваших знаннях, а в вашому прагненні вийти за рамки того, чому вас вчать в школі, чому вас вчать в університетах. Знаєте, на четвер буде по лідерству лекція дуже цікава на Київському економічному форумі, і її буде вести, до речі, запрошую. З одної сторони, власник BlaBlaCar, який з маршруток зробив $1,6 мільярда. З другої сторони, відомий боксер Усик. І я до чого це кажу? Що лекція по лідерству говорить про те, що не потрібні якісь окремі знання. Потрібно окреме бажання бути найкращим і винайти щось більше, ніж те, що вже знайшли до тебе. І сьогоднішній Нобелівський лауреат, вже 4-й Нобелівський лауреат, якого ви і ваші колеги бачать в цій залі, і всі вони без виключення кажуть, не стійте в рамках тих знань, які ми вже вас донесли, не бійтесь експериментувати, виходити за рамки і пробувати доказати те, що на перший погляд здається неможливим. І це той шлях до успіху, який робить вас особливим. Вам успіхів, кожному із вас бажаю досягти чогось особливого.
Дякуємо. А ми запрошуємо всіх до спільного фото. Прямо тут воно буде, ви так, зійдіться до середини.